Okay, this is part 3 of the infection and response revision video. We'll do one more question. We'll do question 5, which is a student is given a tube containing a liquid nutrient medium. This medium contains one type of bacterium. A liquid nutrient medium is it's a liquid that is rich in nutrients, and it is used to grow bacteria. And when we say grow, we mean culture bacteria. In the question, it will be assist on good English, organising information clearly, which is always one for the longer answer questions, and then using the specialist terms where appropriate. Just before we start, just to highlight that when we talk about growing bacteria, when we grow because our 1 cell turns into 2, 2 cells turn into 4, 4 into 8, and that's how we grow, our muscles grow, our bones grow. But that's not quite what we mean when we talk about bacteria growing, because remember, bacteria are unicellular. So that means when we grow bacteria, it's not in size, but in number. So bacteria grow in number. So we call this where we've multiplied the bacteria has multiplied from the 1 to many. We say this is a colony of bacteria in the Petri dish. So the student is told to grow some of this bacteria on agar jelly in a Petri dish. A Petri dish is what we use to grow bacteria in. It's a flat dish with a lid, and agar jelly is another type of nutrient medium, not liquid. It's a jelly medium, and again, it's rich in the nutrients that bacteria need to grow. For this question, we need to first describe how this student should prepare an uncontaminated culture of bacterium in the Petri dish. And to get our 6 marks, we need to explain the reasons for each of the steps that we describe. So it's a 2-part question again. We first need to describe what it is we are doing, and then describe what it is we're doing, and then explain why we are doing this step to make sure it's an uncultured, uncontaminated culture of the bacterium. So how is the steps that we are taking going to make sure that this bacteria is not contaminated, is still sterile? So again, to organize your information, we're going to first talk about, so we're going to organize your information. I'm going to start by, because it talks about steps, I'm going to number my steps so I don't forget anything important. Step 1. First thing we're going to do is prepare this Petri dish. We need a Petri dish with the agar jelly. But to make sure that, again, everything is uncontaminated, we need to make sure that the Petri dish with agar is sterilized before being used. This might come in a sterile, so when you buy it from the lab, it's come in a sterile packaging, or you've bought it and you're going to make sure that it's sterile yourself in the lab. So that's me describing. Also need to explain why do we need to make sure that this Petri dish is sterile. I'm doing it in red pen so we can really see where the described parts are, where the explained parts are. To make sure unwanted bacteria, unwanted because we are growing bacteria, the one that we want to study, but we don't want other strains, other species in there. So we sterilize this Petri dish to make sure that this unwanted bacteria are killed and won't contaminate our sample that we want to study. That's step 1. Then you'll remember that we use something called a inoculating loop. So this is what an inoculating loop is. It's got a loop end over here. That's the end that we dip into the bacterium, and we'll use this to streak our Petri dish. But again, we need to make sure this is sterile too. So to make sure there's no bacteria on there, you will pass that through a flame. So we're going to pass the inoculating loop. So this inocu, and this is where the use of specialist terms comes in. So we're not just calling it a loop or the stick or whatever, but we actually remembering that it is called inoculating loop, and this is passed through a flame. That's our second step. Me describing what the second step is, now I'm going to explain why. To kill unwanted bacteria on the loop. Any bacteria on this loop, passing it through the flame has killed it. That means that this is now clean too. Step 3 is sterile inoculating loop is now going to be dipped into the liquid medium that's got the bacterium in there, and going to use it to streak bacterium on agar dish. Streak is another specialist term. When we streak bacteria, you'll remember that where you've got your Petri dish, streaking where we first streak the bacteria with your inoculating loop in this direction, then this, and lastly like this, and in this area here is where we will be able to isolate the colonies of bacteria. So that's the streaking method. Step 4. When you do this, the Petri dish lid is opened as little as possible. So you don't completely take the lid off, you're just lifting the lid up on one end and then streaking your bacteria. That's the describe. To explain. To further prevent microbes entering the dish. And then the last step, I'm going to put turn the page because there's no space on that one. Step 6. This Petri dish is placed in appropriate conditions and is now incubated. And we do this, we incubate the bacteria in those appropriate conditions where we're talking about some might need an appropriate level of moisture, temperature, for example. We do this because we need to now allow bacteria to grow. Those are our describing marks, and in red, we've got our explain marks. We will need to do both to get all 6. A simple listing of your steps won't get you all 6, maybe 3 or 4, but to get all, you need to make sure you're answering both parts of your question.